We're very pleased to have Eric Woodcock with us today from Wayne State School of Medicine to talk about neuroimmune signaling and substance use disorders. Particularly relevant for us at the Pan-Yale PET P30 because we have projects that are actually ongoing looking at inflammatory processes and substance use disorders, especially OUD. And as you all know, neuroimmune and neuroinflammatory processes are getting a lot of press these days, not just for the classic neurodegenerative disorders where they've been featured, but much broader: depression, schizophrenia, and substance use disorders. And so we're excited about that possibility and hearing more from Eric about that. Eric received his undergraduate training at Wayne State University Translational Neuroscience program, and then he did a postdoc at Yale with people that we know well, Rich Carson and Kelly Cosgrove as some of his mentors. And in May of 2021, he's recruited back to Wayne State as an assistant professor tenure track in the Department of Psychiatry and Behavioral Neurosciences. And he has a current R00 that's focusing on PET neuroimaging and opioid use disorder patients. But I think today he's going to focus with us on some of the work that he's already done. So Eric, let's plunge in and hear about neuroimmune signaling and substance use disorders.
Thank you. Thank you very much for that introduction, and thank you for having me. As I mentioned, those who've signed on later may not have heard. Please interrupt me at any point if you have questions. I don't mind addressing them as they arise, or if you prefer to wait to the end, that's fine as well. We are going to be talking about neuroimmune signaling. It is a very hot area. There's a lot going on, and there's still a tremendous amount of work to be done to better understand how this influences addiction. So with that, let's jump right in, if I can advance. There we go. Okay, so first of all, you all, being that it is a foci of your center grant, already do care about neuroimmune signaling, but I'm going to briefly introduce the system and why we should care about neuroimmune signaling in addiction, and then talk about how we can image it using PET imaging, and quickly summarize the literature across substances sort of broken down by category. And I'm going to go into some detail about some potential caveats or possible biases in the studies that have been conducted so far, and what work remains to be done to really understand if the neuroimmune system is perturbed in any of these drug classes. And then finally, I'll talk about some of the work that I did with Drs. Carson and Cosgrove at Yale during my postdoc, and the potential role the neuroimmune system has in opioid use disorder. Okay, so the neuroimmune system, as we all know, microglia are the brain's resident immune cells. They initiate and maintain neuroinflammatory responses, including the sort of stereotyped morphological transformations that occur that facilitate a number of the processes, including phagocytosis and other things. In addition, microglia signal other microglia and other immune cells via cytokine and chemokine secretion, and astrocytes are known to play a role in the brain's immune system as well. So why should we care about this in addiction? Well, just in general, acute neuroinflammation has been shown to alter a host of cognitive processes as well as influence mood, pain sensitivity, bless you, and alter things like motivation. So just in general, acute inflammatory signaling in the brain can alter a host of processes that can collectively influence whether or not a patient relapses. And of course, if acute neuroinflammation is not resolved and becomes chronic, that is a case in which it can become pathological and is kind of the more widely studied role of neuroinflammation in neurodegenerative disorders. So that can also be the case in substance users such as alcohol use disorder. So specifically in addiction, why should we care about the neuroimmune system? Well, if we look at the three-stage model described in Kuv and Volkov, there's evidence for the neuroimmune system across all three of the stages, in that neuroimmune signaling has been shown to be related to things like drug craving, drug-seeking behavior, as well as after an acute drug administration in the development of conditioned place preference and drug-induced dopamine release, as well as when drug is no longer on board and the individual is in sort of that negative affect withdrawal state that Kuv has talked a lot about, where pain sensitivity can be heightened and withdrawal symptoms can be amplified as related to or as influenced by the neuroimmune system. So across all of the stages of addiction, neuroimmune signaling is playing a role or has been shown to influence those factors. And so really there's a lot of reasons to target the neuroimmune system, which is probably why it's such a hot area of research and why so many people are focusing on it right now. So how do we image it in patients? Well, one approach is with positron emission tomography, or PET imaging. And indeed, that is the most widely used approach to study the neuroimmune system in substance use disorders. One tracer, the tracer that we use a lot at Yale, is PBR28. PBR28 binds to the 18 kilodalton translocator protein, or TSPO, and that PET TSPO imaging with any number of tracers, there are several that exist, is the most popular way of studying the neuroimmune system in patients, including in substance use disorder patients. TSPO is expressed in microglia. That said, we know it is also expressed in other cell types. It's expressed in neurons and endothelial cells, which does complicate the interpretation of our neuroimaging data. That being said, it is still the most widely used approach, and despite these caveats, the best understood for trying to understand the neuroimmune system in patients. We know that TSPO availability is increased after pro-inflammatory challenges, including a number of different stimuli, some that we've studied at Yale and others have studied elsewhere. Also, TSPO levels can be decreased with suppression of microglia. So there is a dynamic range of TSPO availability, suggesting that in patients relative to controls, we should be able to detect levels when TSPO is higher as well as lower than our matched control subjects. And indeed, that is the case in the data I'll show you. The gold standard for using PET TSPO imaging is to genotype individuals. There's a SNP that influences binding affinity, and low affinity binders, or labs, should be excluded from analyses as they exhibit negligible specific binding. Additionally, the high and medium affinity binders, that should be included in analyses, should be entered as a fixed factor in analyses in order to accurately quantify. Another critical factor and complicating factor of this particular type of imaging is there is no reference region in the brain for TSPO. There is no area devoid of TSPO. So you have to sample from the arterial in order to get an input function that can be used to quantify TSPO availability in the brain. That is a very technically challenging and also cumbersome process, and something that is is not ideal for both patients and for PET imagers. The metric of interest, or the preferred metric of interest, is total volume of distribution, which we refer to as TSPO availability, and reflects the number of binding sites that are available to be bound by the tracer. And when quantified in this way, we've previously shown at Yale that test-retest variability is pretty good, 7 to 9% across brain regions, suggesting that if differences do exist with a moderate sample size, we should be able to detect them. One other caveat of VT is that it reflects both specific and non-displaceable binding. So we're measuring both the specific binding of the tracer to the target, but also tracer that's just floating around in the brain parenchyma that is not necessarily bound to the target. It is a sort of compound metric in that way, and that is another factor that complicates interpretation of these types of studies. Okay, so all that background to get us to the point where we can start talking about different substance classes. So what I'm going to try and do in this talk is introduce you to the different classes of drugs, talk about what's known preclinically and post-mortem, and what that might suggest for what we would see in patients in vivo. And then I'm going to summarize the PET studies that have been done in vivo and highlight the strengths and weaknesses of each of those studies. So it's known that cigarette smokers recover more slowly from infection, suggesting an immune component, and that nicotine suppresses inflammatory processes. But people are not smoking nicotine, they're smoking tobacco, which includes a whole host of toxins and irritants, which may cause pro-inflammatory signaling in the brain. And indeed, preclinical evidence suggests that there is an upregulation of mRNA expression for pro-inflammatory cytokines. But interestingly, there's not evidence for upregulation of glial markers using immunohistochemistry. So it's possible that there's a dissociation between these pro-inflammatory cytokine levels and what's actually happening to glial cells in the brain. So in people, the initial studies were done at UCLA in Arthur Brody's group, and he showed a rather interesting finding that was not necessarily expected at the time, that smokers actually exhibited lower levels of TSPO than non-smokers, and these were pretty good-sized PET studies, 15 non-menthol and 15 menthol relative to 15 non-smokers. And they saw about a 17% lower levels of TSPO, which was sort of counterintuitive at the time. But one factor to note with these studies is that the UCLA group, they did not sample from the arterial, and so they did not quantify TSPO-VT. Rather, they quantified what's called SUV, or standardized uptake value, which is not the preferred metric because it fails to account for differences in the plasma, differences in tracer levels in the plasma. So the amount of tracer actually being delivered to the brain might differ between subjects, which could ultimately bias what we're seeing in the brain. So despite these rather interesting effects showing lower levels in these sated cigarette smokers, this metric of SUV is thought to reflect more of distribution and availability of the tracer in the brain as opposed to specific binding or TSPO density in the brain. They followed that study up with a second study that was slightly larger. This time smokers were overnight abstinent, and they found remarkably similar effects. So again, lower levels of TSPO, but again, they used the SUV metric. So it's still a little bit unclear whether or not these are more reflecting tracer distribution as opposed to TSPO availability in the brain. Both of these studies were, groups were really well matched. They did a really nice job matching groups, and all the experimental details were very well controlled. So all afternoon scans, smokers were either sated or overnight abstinent. So all other factors were really well done. But unfortunately, they don't have the capability of sampling from the arterial at UCLA, so the PET imaging metric is not the optimal TSPO-VT. So a study by the Yale group, and specifically Ansel Homer and Kelly Cosgrove, showed in 20 smokers and 20 non-smokers, so a slightly larger study, no differences in TSPO-VT, and this they used the arterial input function. So again, this highlights kind of the importance of using the correct PET outcome for this tracer, because there could be differences in plasma levels getting to the brain. So this study showed no differences, and you can see from the Cohen's d of a 0.09, which is a very small effect. So it's unlikely that even with a very large sample, differences would emerge based on these findings. One caveat for this study, they did not control for smoking recency. Some subjects were overnight abstinent, whereas others had smoked as recent as 2 hours before the scan. So that is one factor that could influence these findings and should be replicated in a future study. On to our next drug. So cannabis is tricky. As we know, cannabis is a complicated plant. There are a lot of phytocannabinoids in the plant material that differ by strain and can greatly influence the effects on the brain. So I've done my best here to sort of summarize what the literature is suggesting. That being said, this is still very much an open area of research, just given the complexity of the different strains of plant material and the potential influence of THC effects versus cannabidiol effects versus other phytocannabinoids. So acutely, research seems to indicate that there is an anti-inflammatory effect, whereas with chronic cannabis administration, there is an upregulation of microglial markers in rodent models, as well as higher pro-inflammatory cytokine levels in human cannabis users. So suggesting that in chronic users, we might see an upregulation of TSPO, suggesting neuroinflammatory signaling relative to controls. And that is exactly what we see in the one study that's been conducted so far. So De Silva et al. In 2019 published that higher levels of TSPO availability, and they did quantify the preferred VT metric, were elevated by over 20%, or about 23% on average, across regions of interest compared to controls. And this was a really nice study. They did a good job matching subject groups for all things except for gender, but they controlled for gender in their analyses. The one caveat with this finding, they did not verify overnight abstinence for their cannabis users. They simply asked them to remain abstinent overnight. So it is one potential caveat, albeit a small one. And then they did subgroup analyses from the same study showing that individuals who met criteria for cannabis use disorder exhibited even higher levels of TSPO than controls. So suggesting that there's almost like a dose-response relationship for the amount of cannabis use and the upregulation of neuroimmune signaling, at least as indicated by TSPO-VT in these subjects. And again, the HABs and the MABs, that's the high affinity binders and the medium affinity binders, so separating out the genotypes. The analyses incorporated them as a fixed factor, but they're just showing you separately. Okay, so for alcohol, alcohol again is also complicated, but for different reasons. Alcohol is such a small molecule that the mechanisms are not well understood, and it's actually thought that a lot of the neuroinflammatory effects of alcohol might be indirect, not from alcohol binding something specifically, but from the damage it might be doing to other systems in the body. So postmortem analyses suggest upregulated markers of glia, as well as it's often cited that neuroinflammation is likely playing a role in the neurodegeneration that's seen in chronic alcohol use. So this has been implicated for several decades, actually, that neuroinflammation may be upregulated in alcohol use disorder patients. Again, the mechanism is not clear, but it's thought that it might be indirect via the quote-unquote leaky gut, where bacteria from the intestines might actually be leaking into systemic plasma, and that's evoking an upregulation of pro-inflammatory signaling, which might be getting to the brain. But that's an area of open investigation. A non-human primate study showed that a single binge dose of alcohol robustly increased TSPO-VT, and that single dose of alcohol, TSPO levels remained elevated for as long as a year. So a very robust acute increase and prolonged increase from just that single administration. Again, suggesting that what we might expect to see in people is an upregulated level of TSPO relative to controls. But much like a lot of things in science, we actually see the opposite. So the initial studies in alcohol use disorder were conducted in the UK by Nicola Kalk, and they showed that a very small sample, only 9 AUD patients, versus 20 controls that early abstinence was associated with lower levels of TSPO than controls. However, the only region that was statistically significant was the hippocampus, as you can see. That study had a number of caveats, unfortunately. While they did calculate VT, groups were not well matched for demographic factors, ancillary substance use like cigarette smoking, nor were they matched for psychiatric comorbidities, which have been associated with differences in TSPO. So a follow-up study by the NIH group looked at, again, early abstinence AUD patients, less than 7 days since their last drink, looking at TSPO levels across brain regions, and they saw no difference in their full sample of 19 AUD versus 17 controls, but in a subgroup analysis, only of medium binders, so excluding the high affinity binders, they saw an upregulation of TSPO levels, or I'm sorry, lower levels of TSPO relative to controls across brain regions. But again, this is a subgroup with only 8 AUD patients in the sample and only 6 controls. So very, very small n's, but suggestive that TSPO levels are lower in AUD, not higher as we had thought. Important caveat of both of these studies is that these patients underwent detoxification using benzodiazepines. So benzodiazepines have been shown to influence TSPO levels, and so it's possible that there are some medication effects for some of these patients which could be influencing findings, but nonetheless, the initial 2 studies suggesting lower levels of TSPO in AUD patients. A study at Yale also looked at AUD subjects and found similar effects, lower levels of TSPO compared to controls. The only region that they saw was significant was in the cerebellum. So again, these are sort of subtle effects, about a 10% difference, and they might be isolated to specific regions of interest. A follow-up study, though, for that, from those data, suggested that the VT differences that they saw in the cerebellum were actually not attributed to specific binding for the tracer, but attributed to the non-displaceable component of VT. So again, further complicating findings, possibly suggesting that there may not be differences in specific levels of TSPO, but rather that it's something to do with how much tracer is getting to the brain in AUD patients. Okay, so this is a lot, I know. This is the last class of substances, and then I'm going to give you a little summary of what we know to date, and then we'll go into opioid use disorder. So in stimulants, specifically cocaine and methamphetamine, postmortem analyses indicate higher levels of glial markers, so suggesting that we might see higher levels of TSPO in patients. And this is consistent with the acute challenge data, suggesting an upregulation of microglia. Likewise, an acute challenge of methamphetamine has been shown to increase microglial markers in rodents. Interestingly, though, the findings are actually mixed in postmortem analyses of methamphetamine users, suggesting some studies find no difference in upregulation of microglia and some do. So it's not exactly clear what we might expect to see in patients. The first study I'll show you in cocaine use disorder was from the Pittsburgh group and Raj Narendran. This is a really nicely done study. These individuals were abstinent for a little over 2 weeks. All cocaine use disorder patients, they were very well matched with the controls. And what you're seeing on the left are high affinity binders relative to controls, and then on the right are medium affinity relative to medium affinity controls. And as you can see, there's no difference between groups, and there's not really even a hint of a difference for any of the ROIs. And this was a really well-matched study, and they quantified TSPO-VT, suggesting that cocaine use disorder in abstinent users is not associated with disruptions to TSPO. Likewise, in methamphetamine users, this time they're active users. it's a very small sample, only 11 active users, but again, no differences and not really even a hint of a difference except for the hippocampus, where there's sort of a possibility of a trend level effect indicating that methamphetamine users may actually exhibit lower levels relative to matched controls. The UCLA group and ED London looked at this in controls and methamphetamine users, again using the SUV metric as opposed to TSVO-VT, which we would prefer, but they found no differences between groups, and the percent differences were rather small for methamphetamine versus control subjects. So all the evidence in stimulant users seems to suggest that neuroimmune signaling is actually not disrupted in these individuals. So I've shown you a lot of data and kind of summarized the whole literature. So I'm going to kind of go back through everything and show you where we're at. So the one study that calculated VT showed no difference in smokers between controls, and that study had well-matched groups. In cannabis use disorder, there's been one study that published TSVO-VT, and they do show an upregulation in cannabis users relative to control. Alcohol, there's been a number of studies, each of which has had interesting caveats and challenges to interpreting the data. The non-human primate literature suggests that an acute binge level amount of alcohol robustly upregulates TSPO, and there's a clinical translation of that study is happening at Yale, and the initial findings, unless something has changed recently, were that they're seeing similar effects in humans. However, in alcohol use disorder patients in the chronic condition, TSPO levels may actually be lower in AUD than controls. Again, albeit in the various studies that have been conducted so far, each of which had sort of interesting quirks or caveats with each. So it's not really clear what's happening in AUD, but there's sort of a hint that AUD patients may exhibit lower levels of neuroimmune signaling. And then finally, stimulants. There's no evidence for differences for either cocaine or methamphetamine users. So at this point, I'd like to stop, and if there's any questions about what I've shown you so far, I'm happy to take them now, and then we'll jump into opioids after this. Okay, let's go on to opioids. Okay, so this is some of the work that I did during my postdoc, and then we're going to we'll talk about some future directions for this research. So with opioids, cellular and preclinical studies suggest that acute opioid administration evokes an immediate and short-lived pro-inflammatory response, and this has been shown with pro-inflammatory cytokine levels and chemokine levels, as well as with immunohistochemistry, or immunofluorescence, sorry, for microglia and for astrocytes in the central nervous system. So both in the periphery and in the brain, there's evidence for these acute but relatively short-lived pro-inflammatory responses, followed by an undershoot that can last for up to several days. But what about in the chronic condition with repeated opioid administration in patients? So preclinically, the evidence suggests that repeated opioid administration can prime microglia and actually lead to microglia cell death. So whereas a single or several administrations can be pro-inflammatory and activate microglia, chronic administration can actually cause them to die off. Conversely, in post-mortem analyses, we see the opposite. OUD patients exhibit higher levels of microglia markers than matched control subjects. Now, obviously, that's post-mortem, and it may not reflect the in vivo condition, but that's sort of the evidence to date. Now, what do we know about OUD patients that might be coming into the clinic? Well, we know from decades of research that they exhibit peripheral immunosuppression, which makes them more susceptible to infections. Infections last longer, are harder to treat, etc. But we actually don't know a whole lot about what's happening in the brain. And in fact, there have been no PET TSPO studies published to date. So the work that we have done thus far, and that I'm going to tell you about, we conducted an acute study where we challenged with morphine to see if we could see an immediate neuroimmune response as evidenced by increased TSPO levels acutely, and we conducted that study in healthy adults. We then investigated what's thought to be the putative mechanism of action for opioids' neuroimmune effects, namely binding to the toll-like receptor system, and specifically toll-like receptor 4. So I'll tell you about that study in non-human primates. And then finally, we did a very well-controlled inpatient study looking at OUD patients on an individualized dose of buprenorphine versus matched control subjects. And we really went to great lengths to try and match them as well as we could on as many factors as we could. All of this is preliminary data, so please do not distribute or copy. We are in the process of writing these up, but they are not yet published. So this first study, we recruited 10 healthy volunteers, and these people were very healthy. They had no DSM-5 diagnoses, no medical contraindications. They had previously been exposed to opioids for medical purposes. They had never experienced an adverse reaction to opioids. They had never used recreationally, and they had no family history of OUD or of AUD. So again, very, very healthy. And then we had every reason to believe that challenging them with morphine would be safe and would not cause them any significant harm. And indeed, we followed up with them and it did not. This was a single-day study. So they came in the morning and did some behavioral testing. Then they had a baseline PBR28 scan. We challenged with morphine, and then we repeated the behavioral testing and the PET scan. We tested 2 doses of morphine, a low dose and a high dose, 0.04 mg per kg given in the shoulder, intramuscular, and a 0.07 mg per kg dose also in the shoulder. The high dose did cause nausea for 2 subjects, and thus they did not complete the post-scan. So we have 4 completers in each the low and the high dose. For you PET imagers in the crowd, we did not see differences between the pre and the post morphine scans for things like dose, mass, plasma-free fraction, etc. We did see a reduction in the arterial input function, which is accounted for in our analyses, but that's also consistent with what we've seen previously in our other pro-inflammatory challenge studies. Things like lipopolysaccharide are associated with a decrease in the arterial input function that we've seen actually a number of our studies. All right, so without further ado, this is what we see. So we have regions of interest plotted, TSPO-VT. The high dose individuals are in the blue lines with the square markers, and the dashed orange red lines are for individuals who receive the low dose of morphine, and the mean for each condition is in the thick blue and the thick orange line, respectively. So we see a robust increase in TSPO availability across regions of interest. Every region was statistically significant, and the high dose condition was associated with a 28 to 39% increase, and the low a 19 to 32% increase. We did not see a statistically significant dose response effect, which is actually due to one subject. If you look at high_1, that subject did not exhibit a response at all to morphine. We are not exactly sure what happened with that person. We think it's possibly a genetic polymorphism that makes them relatively immune, quote unquote, to the immune effects of morphine. But that person was the only person that did not exhibit a significant increase in TSPO levels, and all other subjects exhibited an increase that is beyond test-retest variability. So we had 1 non-responder, 7 of 8 exhibited robust responses. But nonetheless, overall, we see a statistically significant increase. That's a p of less than 0.001. And here are some pretty pictures to sort of summarize that graph. These are mean parametric images for the low and the high dose groups. So the mean of the 4 subjects in the low dose on the top row, high dose on the bottom, pre-morphine to the left, post-morphine to the right. And you can see with the increase in orangish reddish colors, an increase in TSPO availability. And what's particularly interesting and consistent with what we hypothesized, that the bulk of the effect is in gray matter regions. So you can see that there's especially orange reddish around the cortex where we think microglia and potentially toll-like receptor 4 receptors are especially dense. Where they are less dense in white matter regions and along tracks, we see much less of a response. So that was actually really nice to see, consistent with what we expected. As you can see, morphine, or as you might expect, morphine evoked a subjective high. This subjective high tended to peak right around 60 minutes after the dose. We did not see a statistically significant increase in nausea, which was kind of interesting because we did have 2 subjects who were included in this analysis who did get quite nauseous, but nonetheless, it was not statistically significant. And what's really interesting and an absolutely fascinating finding is that what's shown here to the right is a partial correlation controlling for the morphine dose administered, so the actual number of milligrams administered. We see a positive relationship between subjective high after morphine and the neuroimmune response in the caudate. So a bigger neuroimmune response was associated with experiencing a greater high for these individuals. That suggests that neuroimmune signaling may play a role or may be related to the experience of subjective opioid high. We did follow this up. The caudate was an a priori region. We followed this up and tried to look at the ventral striatum, specifically the nucleus accumbens. TSPO is a bit of a noisy tracer, so there is a lot of noise with it, especially in small regions. And unfortunately, in the accumbens, this relationship is not statistically significant. The P, I believe, was 0.11 maybe. It was right around a trend level, but it does drop off for that smaller region, which is unfortunate. But nonetheless, very interesting, especially given the prominent role of the caudate in appetitive motivation and drug addiction. Also, we collected blood samples at several time points throughout the study, and we sent those in to be for mass spec analyses so that we could look at the concentration of morphine, morphine 3 glucuronide, and morphine 6 glucuronide, M3G and M6G on the graph. Those are the 2 principal metabolites of morphine that are associated with effects in the brain. So morphine 6 being associated with binding the mu-opioid receptor and mediating analgesia, and morphine 3 being related to the neuroimmune effects of morphine, or at least thought to be related to the neuroimmune effects of morphine. So we see dose response effects where obviously giving a higher dose of morphine leads to higher concentrations of morphine in plasma. And what's interesting is we see a linear relationship, a very strong linear relationship between the concentration of morphine in plasma at the time of the scan, or over the course of scanning, and the increase in TSPO-VT in the prefrontal cortex. So this gives greater confidence that what we're seeing in terms of a TSPO response might actually be directly related to morphine's effects circulating throughout the body. So this is really interesting and strong finding, and we actually see it in a couple other regions of interest as well, beyond just the prefrontal cortex. So the implications of these data, these are healthy controls. As I've mentioned, these are not patients. And the the clinical implications of these data, we think, or the strongest case for that, is that not everyone goes to becoming an OUD patient the same way. There are a lot of different pathways to developing problematic opioid use, and one such pathway is actually by medical opioid exposure for medically necessary reasons. As in the case of surgery, it's estimated that between 3 and 10% of opioid-treated patients, people who were healthy prior to surgery, ultimately go on to become long-term opioid users, which puts them at very high risk for OUD. So some percentage of those subjects actually are going on to develop an OUD from their medically necessary exposure to an opioid. And that's where we think these data have their greatest clinical importance, because we know that neuroinflammatory signals are associated with diminishing analgesic effects of opioids, enhancing the development of opioid tolerance and opioid reward. So the neuroimmune signals that we're picking up with TSPO on our PET scans suggest that they're present in these individuals who are exposed to morphine for medical purposes, and that blocking them with something like a glial modulator like ibutylast may actually reduce opioid demand after surgery, because it will enhance the analgesic effects of opioids and reduce the development of tolerance. So it's possible that pre-treatment with something like ibutylast before a surgery, where opioids will be needed for pain management, may reduce the risk of OUD in these patients. Study 2 is a very quick study. We were interested in the potential mechanism of action for these effects, which has been hypothesized to be the toll-like receptor system, as I mentioned. So morphine is metabolized into morphine 6 glucuronide and morphine 3 glucuronide. Morphine 6 is associated with analgesia and the mu-opioids receptor system, whereas morphine 3 is thought to bind toll-like receptor 4 and toll-like receptor 2 and upregulate cytokine secretion, which ultimately leads to the neuroimmune response that we measure on a PET scan. So it's this system in particular that we were interested in perturbing to see if it would affect what we see on our PET TSPO scans. So prior evidence has shown that a host of opioids binds to the toll-like receptor 4 system, and in particular, morphine 3 does, whereas morphine 6 does not. And so that we were able to get our hands on a compound called plus naloxone, which is the enantiomer of minus naloxone, which we all know of as Narcan. Plus naloxone is actually a toll-like receptor 4 antagonist, but does not have affinity for either the mu, kappa, or delta opioid receptors in vivo, whereas minus naloxone does. So we were able to get our hands on this compound and try to test whether or not we could block the toll-like receptor system, which may reduce the increase in TSPO we see on our PET scans. And so this was a very brief non-human primate study. We had 2 adult rhesus macaques complete the morphine-only condition, which essentially replicated the design I just showed you in humans. So we get a baseline scan, we challenge with morphine, 2 hours later we repeat the PET scan. And then we completed 1 additional monkey that also completed the morphine-only condition, but we pre-treated with plus naloxone in that monkey. So that monkey received plus naloxone 10 minutes before morphine, and they were at the same dose of 1 mg per kg. The morphine was given IM to match our human data, then plus naloxone was given IV to make sure that it got everywhere before we had morphine on board. Plus naloxone was provided by Dr. Kenneth Rice at NIH very generously, but unfortunately he did not have enough compound for us to complete 2 macaques in the plus naloxone condition, which is why we only have the 1. And so that's all the data you'll be able to see here, which I show on this screen. So what you're looking at is the percent increase in TSPO-VT, so the post-morphine minus the pre-morphine condition. And what's plotted in dark bars is the morphine-only monkeys, and there are two, so you see error bars. And the one that received plus naloxone does not have error bars. It's only the one macaque. So these findings indicate that under conditions of morphine only, there's a 37% increase in TSPO-VT, so a robust response. And when plus naloxone is on board, we see only a 24% increase. There's about an 18% reduction in morphine's neuroimmune effects when we block toll-like receptor 4. So that suggests that toll-like receptor 4 is involved or may be involved, but it is certainly not the only player in this system, as we still see a 24% increase in that one macaque that received both naloxone and morphine. So it does attenuate, but it certainly does not abolish morphine's effects. So this is the final study, and I think we're doing pretty good on time. So this is the study in OUD patients, and then I'll kind of quickly summarize everything, and then I'm happy to take your questions. So this study is a small pilot study, but it was it's a very well-controlled study, and I'll walk you through it here. So we were recruiting patients who sort of resemble, or as close to resemble, patients that you might see in a clinic anywhere. So we didn't have a lot of exclusion criteria. We tried to have as few as possible to make these data generalizable. We were generally recruiting people who had intact immune function outside of their opioid use. So they were not HIV positive, they did not have acute hepatitis C, and they did not exhibit bipolar, psychoses, or current benzodiazepine use. We did allow them to meet criteria for nicotine use disorder and cocaine use disorder, as those are highly comorbid. We allowed some cannabis use, but we did not allow them to meet either cannabis or alcohol use disorder. A lot of our subjects, in fact, I think all of them reported injection use. I think one subject had stopped injecting in the year leading up to the study, but was still using opioids illicitly. We genotyped them and only included HABs and MABs. So this is an inpatient study. We screened them and admitted them inpatient. They were titrated to an individualized dose of buprenorphine to minimize withdrawal symptoms. So this was to make them as comfortable as possible. And it was not mandated by study. It was really just clinically driven. And then we waited at least 5 days after they were on a stable dose of bup that was doing a good job of suppressing the withdrawal before we put them in the PET scanner. So they were either at steady state plasma levels or they were very close to being at steady state plasma levels at the time of the scan. They were inpatient at least on average 12 days before the scan, with the total inpatient stay of about 16 days. And there was some variability across subjects for that. And then they were discharged to outpatient treatment after the study. We went to great lengths to match our control subjects, our comparison subjects, to our OUDs on all demographic factors, smoking status, ancillary substance use, everything we could to really try and isolate the effect of chronic opioid use on TSPO. And that's reflected in this demographics table here. So we recruited, despite COVID, we were able to recruit and complete 6 OUD subjects for the study, and we had 18 healthy controls. And so we used a 3-to-1 matching scheme. So we found 3 healthy controls that approximated age, sex, weight, etc., to each of our OUD subjects to match on as many things as we could. And that's reflected by the lack of differences between groups across a whole host of factors. And this is what we saw. So what's plotted here is PBR28-VT. OUD subjects are in the solid red circles, and healthy controls are in the hollow blue circles. And ROIs are plotted across the x-axis. And you can see that there is some variability across regions, but in general, it's pretty consistent. And OUD subjects exhibit 13 to 24% higher VT compared to controls, and it was statistically significant in the hippocampus and putamen with trends in a number of other regions. On average, we see about a 20% increase across ROIs, or 20% higher VT across ROIs. And this is, again, consistent with our acute opioid study and sort of how we were conceptualizing neuroinflammatory signaling in patients. Here's the data plotted as a percentage. So TSPO levels percentage higher in OUD greater than controls. And so you can sort of appreciate across ROIs, we're seeing pretty consistent effects, with the exception of the occipital lobe, which was kind of interesting. The occipital being the region that is devoid of mu-opioid receptors. So it's interesting to see that that level, or that region seems to be a little bit lower than all others. Now, this is just 6 OUD subjects, so this needs to be taken with a grain of salt and obviously replicated in a larger sample. But nonetheless, this is sort of strong preliminary findings suggesting that there may be group differences very early in treatment for patients who we know are stable, we know have not been using opioids, they're in a locked inpatient unit. So this is very strong preliminary data, and we're interested in following up on this. In addition to those main effects, we saw some really interesting correlations. So controlling for genotype, individuals who are inpatient longer prior to the time of the scan, so they had greater experimental abstinence, if you will, exhibited a negative relationship with TSPO levels in the prefrontal cortex. And being that OUD subjects had higher levels than controls, this possibly suggests that with longer abstinence, you might see TSPO levels sort of creep back down to what's more normal and more closely resembles healthy controls. Obviously, that is speculation. These are not within subject data. This is across subjects. So we would need to follow up with a within subject study to really know that, but it is nonetheless suggestive. And then finally, we see higher levels of opioid craving were positively correlated with higher levels of TSPO availability. So more neuroimmune signaling in the prefrontal cortex, possibly indicating greater craving. So in summary, across the 3 studies, I showed you that morphine evokes a neuroimmune response, and that neuroimmune response might be related to experience of subjective high after morphine, and was linearly related to plasma morphine concentrations. There's some initial evidence that toll-like receptor 4 binding might be a mechanism related to morphine's effects on the neuroimmune system, at least as evidenced by TSPO. And finally, in study 3, I showed you that OUD subjects exhibit elevated neuroimmune signaling early in treatment on an individualized dose of buprenorphine, and that longer time in treatment may be associated with more normative levels, whereas higher levels of TSPO are related to higher levels of craving. The clinical implications of this are that glial modulators have been shown to reduce neuroinflammatory signals, and in preclinical and clinical studies have been shown to exhibit a whole host of positive benefits related to addiction processes in opioid use disorder, such as reducing opioid-seeking behavior, opioid craving, and opioid withdrawal symptoms. So it's possible that glial modulator medications could be beneficial for some patients. And then I'm just going to throw up the future directions slide quickly. These are some areas that we are hoping to pursue in the future to really better understand this system and how it might be useful for informing treatment, and specifically whether or not neuroimmune markers actually predict treatment success, whether they do exhibit normalization with prolonged abstinence, and whether glial modulators may be effective adjunctive medications for OUD. And with that, I'd like to thank my collaborators at Yale, my funding at NIDA and NIMH, and you for your attention. I'm happy to take any questions. I think we have about 5 minutes, and I can stick around after 4 p.m. if people have additional questions.
Hi,
jump in,
this
Eric,
is
and say that was terrific. Go ahead. Anybody who has a question?
this is Toby Eisenstein. That was a really interesting study. I'd like to focus on the part about opioids binding to TLR4.
Hmm.
I've written a critical review looking at the literature,
Hmm.
basically based on the fact that virtually the entire literature on opioids in the periphery shows that they are immunosuppressive. And
Hmm.
if either morphine or its metabolites bound to TLR4, you would certainly see a pro-inflammatory effect, which no one really has reported. There's also
Right.
been some critical review of the plus naloxone, and a number of the intramural people at NIDA were unable to replicate those studies when the plus naloxone was sent out to 3 independent laboratories. That is in terms of binding to TLR4. So the effects that we're seeing may be real, but I doubt that they're happening through TLR4 binding. And I think that this has confused the literature enormously, and we should be very cautious in taking those conclusions as something that is a given. One alternative interpretation that I proposed is particularly for chronic opioid use, either in animals, potentially in humans, is the thing that you suggested in alcohol use disorder, which is that the drug is actually causing leakage of gut organisms from the gut, releasing LPS into the blood, and the LPS binds to TLR4, and that is what is causing a pro-inflammatory response. And we actually published back in the 1980s that morphine induces sepsis in mice.
Yeah. So I guess first, I certainly didn't verify that plus naloxone binds to TLR4. We didn't do any of that work. We had just the one macaque. We had just enough plus naloxone for the one macaque. So we're, a whole host of limitations and caveats for our single monkey study, right? Certainly can't make any strong claims based off of what we're finding. As you do mention, that has been a big topic of discussion is the TLR4 system. Is it actually mediating these effects? Is it even related to these effects at all? I'm not a cellular or preclinical person, so I don't do any of that research. We're sort of just at the mercy of what the literature is suggesting to us in terms of what we can test. And again, a single macaque is not conclusive of anything, and the effects we saw are actually relatively small. 18% reduction is certainly not indicative of complete abolishment of that effect. And lastly, in terms of leaky gut, absolutely. So as I mentioned, all of these individuals had reported injecting opioids at some point in their lifetime, and I believe 5 of the 6 were current injectors. So it's very likely that the stuff they're putting in their veins is not pure. It's got all sorts of crap in it that's causing an inflammatory response that could be completely separate from any opioid effects. And that could explain why we're seeing such a strong relationship between days inpatient and essentially days abstinent and the reduction in TSPO levels. That it could simply be due to the fact that they're not putting whatever bacteria and other irritants directly into their veins.
Okay, thanks.
Thank you very much. Yeah, it's a really it's difficult because people are making very forceful claims about TLR4 and the role of the opioid system. And I'm a clinical person, so I'm trying to figure out what's going to be useful for patients and what might be useful in the clinic. And it's really difficult to know which direction to go.
Just a follow-up. This is Tom Graham. I don't know if you've considered in addition to TLR4, the MRGPRX2, which is is a G protein that's involved in itch. And so morphine is thought to activate it and cause mast cell degranulation, which then leads to the itch effect. I don't know how it would impact TSPO, but it certainly seems like it's on some kind of immune axis, and I wonder whether or not there's any value to like having a look in that literature. It's been reported by Brian Roth's lab, like 2018, and then there was a paper in Nature recently with this crystal structure. I could
Brian Roth,
send it
is
to
that
you
what you said?
offline.
Yeah, I would appreciate that. Thank you.
Eric, one of the things that was intriguing to me, we're always sort of sensitive to the issues of heterogeneity, which is, sort of unavoidable in our lives and in our disorders. But your genetic parsing of the data is intriguing to me. Do you want to give us a quick thumbnail about your hypotheses and how these high and low affinity, binders, how you came across that and what you're going to do with it? And do you see anything clinically with them that's different?
Yeah, so that was actually out of the UK. Dr. Owen's lab discovered that, and really what it was is we were seeing just huge differences in TSPO availability in healthy individuals who should exhibit relatively tight band of variability, and we were seeing essentially a bimodal distribution. And he ultimately, him and his team, found that there was this SNP that accounts for that variance. As far as clinical outcomes in these groups of people, I don't know. I'm not aware of anything in addiction. I know that there's been some work in depression, looking at just given the role the putative role of the translocator protein in cholesterol import into the cell and then ultimately synthesis of neurosteroids and hormones. That there's been work in looking at allopregnanolone and the influence on depression, and so the potential would be that do these individuals who have higher affinity for this tracer, do they also have higher affinity for cholesterol or any other cofactor that may enhance the import of cholesterol into the cell? And I don't know what the state of that literature is.
But I mean, it's a very elegant tool to have because, of course, you can imagine just talking about all the challenges in the literature, and there's so many in terms of the nature of the tracer, the nature of the patients, how well they're matched, how many other disorders they have, even if they're not using at the level of a disorder, if they've had use, it's tough. It's tough going. So I think that the genetic tool is it's sort of a really appealing thing, and I'm glad you mentioned it, and I just wanted to highlight it a little bit. And to thank you was a real tour de force, the other substances as well. It's now making the opioids look a little bit more special, isn't it?
Yeah, and I think further just to highlight the fact that it's we often think about these things as the drug that they are using as the only pharmacologic agent that's on board, but in the case of opioid use disorder, especially with injection users, they're putting all sorts of crap into their bodies, and we don't know what it is. So there's all sorts of other things that are influencing the immune system beyond any effects that may be happening with the opioid itself. And in this day and age, it's almost exclusively fentanyl, depending on where you are. So it's probably not even heroin
Right.
going through any of the morphine system for its metabolism. So it's a very complicated. This tracer PET TSPO is far from perfect. It's incredibly complicated to interpret. It's incredibly complicated to conduct these studies. So we're only sort of beginning to scratch the surface for understanding what might be happening in our patients.
Well, you've done more than scratch the surface today. Do we have any other questions from folks? I know it's a little bit after 4. And so if some people do have to leave for clinical or other duties, they can do so. But I wanted to give people a chance to ask questions that might not have yet had a chance. The other thing is, if you have something, I'm sure Eric would be happy to answer by email if something comes up afterward that you think of that sort of hits you in the middle of the night and you sit up straight in bed and say, I should have asked this, I'm sure you'd be willing to answer. So we'll try to inhibit that. We won't text you in the middle of the night, but if someone does have a question, they could email you the next day.
I have 2 small children. I'm probably awake.
And thanks very much, and congratulations on the next step in your career, and we'll be following you. Thanks a bunch, Eric.
Thank you very much.
Thank you.